Okay. Chapter 15 feeds off of chapter 14 and 13. So when you're studying 13, you're already starting to study 14 and 15. When you're studying 14, you're reviewing for 13 and you're already investing into 15. Okay. So give yourself time not just to try to memorize information, but to connect the dots, as I call it, to help you out. Okay. Now, chromosomal basis of inheritance. Chapter 14, basically the focus is what they call Mendelian genetics. A monk by the name of Gregor Mendel performed what is now referred to as a foundation for genetic studies with pea plants, studying the patterns of inheritance. He studied a total of 7 traits. Going into the test, you're responsible for 3, flower color, and seed shape. And he basically performed 2 types of experiments, either monohybrid crosses, where he focused on how one trait was passed on from one generation to the other, or dihybrid crosses, where he focused on 2 separate traits, such as seed color and seed shape. And he wanted to know if, from one generation to the next, we only have dominant traits passed on or recessive traits passed on for the different. In other words, are they linked or are they not linked? Is it possible for an offspring to inherit a dominant trait, but for the other different trait, a recessive trait? From these crosses, he came up with 2 laws. From his monohybrid crosses, he came up with the law of segregation. From his dihybrid crosses, he came up with the law of independent assortment. Okay, when you're studying chapter 13, meiosis, specifically meiosis 1, this is where you see these terms, segregation and independent assortment. Both of these happen, okay, during meiosis 1. Oops, God, that was bad. It's a little shaky here. So both of these happen during meiosis 1 here, okay? If you recall, we talked about the law of segregation. Law of segregation has to do with where homologous pairs of sister chromatids, okay, will line up forming the metaphase plate. And then in anaphase 1, they will be segregated. In other words, we will go from a diploid to a haploid state, okay? Well, from his monohybrid crosses, he came up with the law of segregation, okay? And this law, if you look here at these figures here, okay, they're showing you a pair of R alleles and a pair of Y alleles. When you go over this law, only focus on one or the other. Because remember, monohybrid crosses only focus on one trait. So when you read through this law that I gave you for the law of segregation, only look at one pair of alleles, such as these R alleles, when you read that definition, okay? And how this one pair of alleles, okay, R alleles, are segregated during meiosis 1. Now, if you look down here, you can see in one haploid cell, we're only going to have the dominant allele for shape. And in the other haploid allele, we're only going to have the recessive allele for shape. So when you go over this law, remember it has to do with monohybrid crosses. For his second law had to do with dihybrid crosses. So this is where you would focus on both pairs of alleles. Now I'm just boxing off these on this side because they're closer to where the law says, but you would actually box off these also, okay? So when you go over this second law, okay, that Mendel derived from his dihybrid crosses, now you're focusing on 2 separate traits. So we're looking at 2 different pairs of alleles for 2 different traits, okay, and how they're independently assorted. Well, I actually have to box both. When you go over meiosis, we talked about in metaphase 1 how homologous pairs of sister chromatids have paired together, and we call these tetrads, okay? This is where we first use independent assortment. And during metaphase 1, when these tetrads align end to end forming the metaphase plate, what side the maternal and paternal DNA is on for this homologous pair is independent on this other homologous pair as far as what side the maternal and paternal DNA is located on. So over here you can see with this metaphase plate, for this tetrad, maternal DNA is over here, paternal DNA is over here. For this other separate tetrad, you can see the same thing. Maternal DNA is over here, paternal is over here. However, when these tetrads align end to end forming the metaphase plate, what side maternal versus paternal DNA is on for these 2 separate tetrads is independent of each other. So over here, that's what they're showing you. Now we have paternal DNA over here and maternal, okay? But you can see over here, we have maternal on the same side as this paternal. So how they What side maternal versus paternal DNA is for this tetrad is independent of how they line up for this tetrad. Thus, we call it independent assortment. Now, what does that have to do with anything? Well, we could end up having gametes that are only carrying maternal DNA or paternal DNA, or we could have gametes produced that are carrying a combination of maternal and paternal DNA. Therefore, whatever alleles are located on these homologous pairs, that can affect what combination we have, thus resulting in Mendel's F2 generation results, what we refer to phenotypically as a ratio of 9 to 3 to 3 to 1. For every 16 pea plants he counted, okay, 9 of them had dominant shape and color. 1 out of 16 had recessive shape and color, but 3 of them had dominant shape with recessive color or recessive shape with dominant color. In other words, dominant and recessive traits inherited, okay, for those two separate traits. How is that possible? Has to do with independent assortment. Okay, so it takes some time to study this to get this down. So for Mendel, what he came up with is his F2 hybrid crosses was that based on the results from the F2 generation, his second hypothesis for independent assortment proved to be true. But this was for pea plants. Okay, so Mendel publishes his work roughly in 1866. At that time, everybody, at best, scientists had discovered there's a new compound, but they had no idea what it was. Then roughly by the 1870s, a biochemist had discovered some of the chemical components that made up the known compound. By the turn of the century, the 1900s, they had called this compound DNA, and they knew it was made up of 4 bases. However, nobody knew what the function or purpose was. Therefore, when Mendel conducted his pea plant studies beginning in the 1850s and published his work in the 1860s, he was strictly going by phenotypic traits. Okay, so traits having to do with observable features. Some of these traits exist as a dominant phenotype. Some of these traits exist as a recessive phenotype. Okay, such as the trait for flower color. For pea plants, the trait for flower color exists as a dominant phenotype purple or a recessive phenotype white. Okay, Mendel was just going by what he could see. The genetic side of it wasn't added, okay, till after the 1950s, basically, as far as conclusively being proven. So after this third exam, okay, chapter 16 will go over one of roughly 3 different groups of scientists that conducted an experiment that conclusively proved DNA is a compound through which traits are passed on from one generation to the other. But what was known even before DNA was finally identified as a compound used to pass on traits from one generation to the other was that Mendel's law of independent assortment from his dihybrid crosses was not 100% true. It depended on the species you were studying. Therefore, okay, some scientists discovered that when they performed a dihybrid cross with other species, eventually what was found is sometimes genes or alleles for different traits are located on the same chromosome. Therefore, instead of law of independent assortment, it was Mendel's other hypothesis that proved true, law of dependent assortment. This is what we're going to get into right now. We call these linked genes, and this has been found to be true for species as they started uncovering more and more of the genetic information that coded for the different traits in different species. What they basically came to find out, okay, is when they look at a homologous pair of chromosomes, such as in human beings, all right, at different gene loci along each homologous chromosome, you will have pairs of alleles that code for different traits. Therefore, say this red maternal chromosome, if it's passed on to an offspring, not only is this allele for one trait being passed on, but automatically these other 2 alleles which are located on this chromosome are also going to be passed along. So in this case, we would have 2 dominant alleles for 2 different traits and 1 recessive allele for a third trait. They would all get passed on together because they're all located on the same chromosome. Therefore, we refer to them as linked genes. Okay. The person credited with identifying linked genes we're going to talk about is a man by the name of Thomas Hunt Morgan. So therefore, linked genes do not follow Mendel's law of an assortment. Okay. Instead, what it is linked genes follow his other hypothesis, dependent assortment. Now, the man credited with discovering linked genes, although he was going by phenotype, and not only was he going by phenotype with a different species, But he also did not believe DNA could ever be the compound through which traits are passed on. He was one of those that was in a large group that believed it was proteins that allowed for the transfer of traits from one generation to the other. Thomas Hunt Morgan, he won the Nobel Prize in 1933, but you can see here he was already publishing work by 1915. He was working with what we call in fruit flies. In lay terms, we call fruit flies. Their technical name, which is in your notes, and make sure you know both, the lay term and the technical term, is Drosophila melanogaster. He was actually studying how traits are passed on from one generation to another in these fruit flies called Drosophila melanogaster. And what he was focused on initially was eye color. Okay. He performed monohybrid crosses to determine how the trait for eye color was passed on. And what he discovered was the red eye color was the dominant phenotype and the white eye color was the recessive phenotype. Later on, geneticists came along and found out that when you look at a population, a group of the same species, or a group of Drosophila melanogasters, you find that not only is the red phenotype the dominant phenotype, it's the most common trait in that population. Therefore, when you take genetics, you start off with Mendelian genetics, which is basic genetics. Whatever the dominant phenotype is, such as purple flower color, we're going to take that first letter and use that to name the alleles that code for flower color, such as uppercase P allele. But geneticists actually have added on to that. Then they'll say, okay, there's a dominant allele and a recessive allele for two different phenotypes, but which trait is more common? So therefore, what they found for fruit flies is that the red eye color is not only coded for by dominant alleles, but it's the most common allele. Therefore, the red eye color is the most common trait. A geneticist will let you know this by calling it the wild type. So red eye color is a wild type. That means it's also the most common trait. The white eye color is actually coded for by a recessive allele. They refer to it as a mutant type. Okay, in other words, this is very rare. Sometimes, and with rare exceptions, sometimes you won't get it so cut and dry where the dominant allele is also the most common type. But I'm not going to get into that. That has to do with population genetics. I'll let a genetics class cover that. So what Morgan did, okay, is basically he took Gregor Mendel's monohybrid cross experiment and he duplicated it using these fruit flies. Okay, so he performed his own monohybrid crosses and he was focused on the trait for eye color for these fruit flies. Okay, if you recall, just like with Mendel, the initial generation was called generation for parent generation. He allowed them to sexually reproduce the F1 generation, their offspring. But for fruit flies, they're animals, so they sexually reproduce. So there's a little more going on, but the bottom line is he did take the F1 generation and he had them produce the F2 generation. Now, if you look at Mendel's work with pea plants and his monohybrid crosses, it doesn't matter if you were looking at flower color, or seed shape. The F2 generation always had a phenotypic ratio of 3:1. You would see for every 4 offspring, 3 of them would exhibit the dominant phenotype and 1 out of 4 would exhibit the recessive. Well, if you look here, Morgan got the same exact results for his F2 generation. On average, for every 4 F2 generation offspring that were produced, 3 out of 4 would have the phenotype for red eye color, 1 out of 4 would have the phenotype for white eye color. We have a 3:1 ratio. So it's working great. Listen carefully. A good scientist has a good eye. Even though they may be focused on a specific type of research for a certain purpose, they have a good eye to where they notice something may be going on off to the side that they originally were not interested in or were looking for. This was the case for Morgan. As he's counting thousands upon thousands of fruit fly eye colors, he starts to notice 2 other traits. Okay, he starts to notice that many of these fruit flies tend to have a certain combination of 2 traits that have to do with body color and wing size. He starts noticing, regardless of whether they have red or white eyes, he starts noticing that most of the time, not always, but the majority of the time, he comes across a fruit fly with a gray body color. The wings have what he referred to as normal development or normal wing size. But he couldn't help but notice that anytime he came across a fruit fly that had a black body color, it tended to have these underdeveloped or kind of ugly looking mutated wings, which they call vestigial. Vestigial just means underdeveloped or malformed. Okay, he couldn't help but notice this combination. So then he said to himself, okay, I'm noticing these two different traits for body color and wing size always seem to be passed on in a certain combination, either gray with normal wing size or black with vestigial wings. So he duplicated Mendel's other type of experiment, dihybrid crosses. Except Morgan performed dihybrid crosses, okay, using fruit flies, and he focused on two different traits, body color and wing size. Again, he started off with a P generation that produced an F1 generation, took that F1 generation and produced an F2 generation. In that F2 generation, unlike Mendel, for a dihybrid cross, the F2 generation should always end up with a ratio of 9 to 3 to 3 to 1. That should be the phenotypic ratio for pea plant F2 generations in a dihybrid cross. But unlike Mendel's results for his pea plant dihybrid crosses, Morgan got something completely different. Just focus on this one down here. Okay, because I'm not going to test you with all this other detail here, just here. Okay, what Morgan, the results Morgan obtained, okay, did not agree with Mendel's dihybrid crosses. When Morgan performed dihybrid crosses with fruit flies focusing on these two traits, his phenotypic ratio came out to what they describe as 1 to 1, 0 to 0. Basically, the majority of the F2 generation offspring either had that gray body color with normal wings or black body color with vestigial wings. He had small numbers that had a combination of gray body color and vestigial, black body color and normal wing. The vast majority, though, fell in these two categories. This went against Mendel's pea plant dihybrid cross. In other words, Morgan proved using a different species that the law of independent assortment is not always true. It depends on the species you're studying. Well, later, as it was uncovered, DNA is a compound, but through which traits are passed on from one generation to another. Geneticists eventually duplicated Morgan's work, but now they were able to analyze the genetic information, okay, contained in the homologous pair of chromosomes that the fruit fly had. And what they came to find out is that they're in the homologous pair of chromosomes, one of the four pairs that the fruit fly has. They came to find out that the allele that codes for body color happens to be located on the same chromosome or homologous pair of chromosomes as the allele, okay, that determines wing size. So what they found out, if you go back, what geneticists found out is that for the majority of those fruit flies that had gray body color and normal wing size, they were actually heterozygous dominant for both types. In other words, geneticists also discovered that gray body color is the dominant type and normal wing size is also the dominant type. Black body color is a recessive phenotype. Vestigial wings is also a recessive phenotype. But when they looked at this F2 generation, they were heterozygous dominant for both. Therefore, they ended up with gray body color and normal wing size. But for the other half, roughly, that had black body color and vestigial wings, what geneticists came to find out is they were homozygous recessive for black body and homozygous recessive for vestigial wing shape or malformed wings. But the problem with that, though, although this proved that alleles for different traits are sometimes passed on together to where if you inherit one dominant phenotype, such as gray body color, you're automatically going to inherit another dominant trait, such as normal wing size, or if you inherit recessive trait, such as for black body color, you're automatically going to inherit another recessive trait, but in this case having to do with vestigial wings. Morgan still had to explain the small number of fruit fly populations which actually exhibited dominant body color with recessive wing shape or recessive body color with normal wing shape. Well, again, geneticists came along and said those two small populations were the results of an event that occurs during prophase 1, crossing over. If you recall in chapter 13, during prophase 1, and you do not see this at any other time during cell division involving meiosis, and you never see it during cell division involving mitosis. There's an event called a crossover or crossing over, which occurs during prophase 1 of meiosis 1. This has to do with where we have a homologous pair of sister chromatids, and when they pair together forming a tetrad, one of these two sister chromatids will cross over with one of these two sister chromatids, and what occurs there? DNA recombination. In other words, they will exchange DNA, and now what happens is they look like this. You can see based on the color scheme, you can see the colors changed a little bit here and here. Now, what does that have to do with anything? Well, during meiosis 2, where the sister chromatids are separated, now what happens is we end up with gametes carrying what you might call hybrid chromosomes. Now, instead of having a chromosome that's only carrying wild type information or mutant information, or another way to say it, dominant information or recessive information, now you have hybrid chromosomes that are carrying a combination of, say, gray body color and vestigial wings, or black body color and normal wing shape. Okay. So eventually, the way geneticists describe this is that when an offspring inherits a phenotype matching just one of the two parents, we refer to the offspring as parental types. But for these two small groups here, these two small groups are referred to as recombinants or recombinant types. In other words, these offspring do not resemble, okay, either parent. They actually represent, if you want to call it, a little bit of both parents inside of them. So we refer to these as recombinant type recombinants, and they're the results of this event called crossing over. Now, I'm not going to give you a Punnett square or anything for this Drosophila or fruit fly experiment that Morgan did. Just know the information I mainly gave you in your notes. But the key point, okay, is that Morgan, okay, did feed off of Mendel's work by performing monohybrid and dihybrid crosses. And for Morgan's monohybrid crosses using a different species, okay, he came to the same conclusion. All right, F2 generation has a phenotypic ratio of 3 dominant phenotypes to 1. However, Morgan's dihybrid crosses contradicted Mendel's dihybrid cross results. Okay, Morgan's dihybrid crosses proved that no, the law Mendel's hypothesis about independent assortment is sometimes true. Therefore, Mendel's law of independent assortment isn't 100% true. It depends on the species that are being studied. So there's variation of the various species that live on this earth. Then the question came about, what about sex chromosomes? Do sex chromosomes carry any genetic information on them? Well, the answer to that was absolutely yes. Okay. We're going to focus on us. Okay. I was trying to get the little pen. Human sex-linked genes. Okay. Here it's showing you how sex is determined in various organisms. What we're going to focus on with us. Okay. I believe everybody should or does know that, okay, if we have a human being that is female offspring, 23rd pair of chromosomes is going to be a pair of X. If we have an offspring that is a male, the 23rd pair of chromosomes is going to be an XY. And so if you're looking at a karyotype and you want to know the sex of the person that they created this karyotype from, you just want to look at the 23rd pair. The X chromosome, if you look down here, the belief is at one time the X chromosome was as large as the Y, if you look at the Y chromosome here, I misspoke. If you look at the Y chromosome here, the belief is the Y chromosome at one time was as large as the X chromosome. Okay, there are some that believe the X chromosome eventually will just vanish. Okay, I feel like that's a little biased there right now, but that would mean the human race would either reach extinction or have to learn to asexually reproduce. But just to let you know, you might hear that sometimes. But you can see there's definitely a difference in size. But they still, as of right now, contain genetic information that's equally important. Now, the Y chromosome is most popular for this gene. The Y chromosome contains what they call a male determining region, or what some refer to as a sex determining region, Y protein. Sex determining region, Y protein, okay, that's what SRY stands for. The SRY gene contains genetic information in it that directs a developing fetus to develop male characteristics. Okay? Therefore, for female offspring, which inherit a pair of X chromosomes, okay, and no Y chromosomes, you can say passively or by default what will happen then is when the Y chromosome is not inherited and instead two X chromosomes are passed on, there's no SRY gene, and therefore the fetus will automatically develop into a female or develop female characteristics. So this has been the key. This is why this gene is so popular, the SRY gene, because it's been proven and shown when this gene is present, the fetus will develop male characteristics. Now, it contains other genetic information, such as for male fertility factors and ribosomal components. Okay. Now, for women, if technically speaking, okay, for female offspring, they have 23 pairs of homologous chromosomes because the 23rd pair of chromosomes are both X. But for males, okay, when you look at a karyotype such as we have here, we only have 22 pairs of homologous chromosomes, and they're all autosomal chromosomes. The last pair of chromosomes, the XY chromosomes, has led to males being referred to as being hemizygotes for the 23rd pair. Okay, so for males, that 23rd pair, because one is an X and one is a Y, males are referred to as being hemizygotes for the 23rd pair. Hemi just meaning half are X, half are Y. The X chromosome itself, okay, I never had a professor say anything about it. I never come across any information mentioning it as far as the X chromosome containing genetic information causing the fetus to develop or promoting the fetus to develop female characteristics, okay? It's there, but you don't hear about that because it's been discovered, or it was discovered, that the X chromosome contains a lot of genetic information that has nothing to do with the sex of the offspring. Okay? The X chromosome has been found to carry alleles responsible for us being able to differentiate between different colors, especially red and green. The X chromosome was found to also contain alleles that code for a blood clotting factor, so that when you get a cut, a blood clot will form and you'll have a scab. The X chromosome has also been found to contain alleles that code for a protein called dystrophin. Dystrophin is a muscle protein found in your skeletal muscle cells, and it basically helps your muscle cells maintain their structural integrity while they're producing movement, preventing them from collapsing. Thus, the muscle cell would not work. Well, genes that code for these different traits are all located on the X chromosome. Therefore, we refer to these as X-linked genes. Okay? These X-linked genes that I mentioned that are associated with differentiating colors, forming a blood clot, or maintaining skeletal muscle integrity. Okay, these are all located on the X chromosome, thus they're linked genes, but also these genes have been found to be tied into three types of genetic disorders that you primarily see occur in males. These three genetic disorders are simply known as color blindness, hemophilia, and muscular dystrophy. Okay. First, what I want to go over is this here. First of all, when we talk about X-linked genes, we first start with the X chromosomes, as I said. Okay. If the offspring inherits a pair of X chromosomes, they're female. If they inherit an X and a Y, they're male. Okay. So when mom and dad are going to have children, if you look here, notice it doesn't matter which of these Punnett squares you're looking at. There's a 50-50 chance they will have a female offspring or a male offspring per child, each child, not combined total, but per child. Okay. When it comes to alleles that code for traits not having to do with sex, what we do is we still write down which sex chromosome we're talking about, and then we put the name of the allele as a superscript. If it's a dominant allele, we'll put something like X uppercase N. If it's a recessive allele, we'll put something like, or we'll write down X with superscript lowercase n. Okay, to denote, hey, we have alleles that code primarily for traits that have nothing to do with sex, but these alleles or genes are located on the X chromosomes. And down here it's showing you, okay? When it comes to the sex of the offspring, it's the father that's going to determine the sex of the offspring. Okay, mom can only pass on an X chromosome. It's dad that will either pass on an X chromosome or a Y chromosome. Therefore, 50-50 chance, one of these two, the sperm will be carrying an X or a Y, 50-50 chance you'll have a male a male or a female offspring. But now look at the alleles. If mom, let's say, is homozygous dominant, okay, for a particular trait, she can only pass on an X chromosome carrying the dominant allele. If dad's one X chromosome is carrying the recessive allele, okay, he can only pass on that X chromosome to his daughter. His son would inherit the Y chromosome. Therefore, when we get into genetic disorders, this is going to be important to note. All right. Now, over here, when you go from here to here, imagine one of these daughters here now is going to have their own child. In other words, they're a carrier. They have the genetic information for the recessive allele, but it's being covered up by the information encoded by the dominant allele. But let's say one of these two here has their own children. Here we have a woman who is heterozygous dominant for this pair of alleles located on the X chromosome, and let's say she has children with this man who actually has an X chromosome with the normal allele. Okay? Again, dad is only going to pass on a Y chromosome to the child. So if mom on one of her X chromosomes is carrying a recessive allele, okay, if the male offspring inherits an X chromosome with that recessive allele, it has to have come from mom because the son is inheriting a Y chromosome from dad. Any genetic information inherited on an X chromosome by the male offspring, it's coming from mom, whereas the daughter's inheriting an X chromosome from both parents. Well, the thing about genetic disorders, okay, is that treating the symptoms has improved, such as hemophilia, bleeder's disease. Okay, it used to be, hey, it occurred primarily in the male offspring. They would be lucky if they lived into their teens. Okay, but eventually what happened is one of the ways to treat the problem is when people donate blood. Okay, not only are the red blood cells isolated, but the clotting factors, these proteins called clotting factors in the person's donated blood, they're also isolated and they're administered to the person who has hemophilia so that they can develop a blood clot. So now what happens is you promote survival, okay, of a male offspring that's carrying a genetic disorder such as hemophilia. You promote their survival. Now, to my knowledge, they're not sterile. They're capable of having offspring. So what's happening now is this is increasing the chance that, say, a male, okay, who has hemophilia, which is a recessive X-linked genetic disorder, could pass on that X chromosome to one of his daughters. And if mom is a carrier, now there's a very slight chance. Okay, it's slight. The probability is very low in actuality, but it's increasing with better treatment. But there's a very slight chance that the female offspring now could inherit a genetic disorder, okay, that for the longest time only occurred in males. You would only see it in the male phenotype. Now it's increasing chances. This is why you hear a lot of talk about systems such as a CRISPR system, okay, and doing performing gene therapy, trying to correct, okay, genetic information inherited by a person. Take that mutated genetic information that's having an adverse effect on a person's phenotype, such as a person with hemophilia can't form a blood clot, trying to fix that genetic information in that person so that now they can start forming their own blood clot and eliminating, say, a recessive allele that causes a genetic disorder. So bottom line is, treatment of the symptoms has gotten better, but the cause of the symptoms hasn't been fixed yet. And this is why for genetic disorders, they want to be able to perform gene therapy and correct those mutated genes. Three most popular genetic disorders, okay, that are X-linked genetic disorders are color blindness, hemophilia, and Duchenne muscular dystrophy. Listen carefully. These three genetic disorders are recessive genetic disorders in female offspring, okay? If a female offspring is heterozygous dominant, okay, What happens is they inherit a pair of X chromosomes, and on one of the X chromosomes is a normal allele for normal, say, normal color vision. On the other X chromosome, they've inherited a recessive allele for color blindness. But what's been found is you only need one good allele, one normal allele. So a female offspring could be a carrier for color blindness, but not have color blindness because on one of the two X chromosomes, they're carrying the dominant normal allele. It's the same case for hemophilia and Duchenne muscular dystrophy. But for males, okay, for males it's different. We only inherit one X chromosome. We're hemizygous. Therefore, unlike for women, where color blindness, hemophilia, and muscular dystrophy are not only X-linked genetic disorders, but they're recessive X-linked genetic disorders in female offspring. In males, since we are hemizygous and only inherit one X chromosome, we don't describe them as recessive. Okay, they are strictly genetic disorders in males. We only inherit one X chromosome. It is a recessive allele that causes color blindness, hemophilia, and muscular dystrophy. Therefore, if the male inherits an X chromosome from mom carrying that recessive allele, then you will see that offspring develop a phenotype that the medical community refers to as being colorblind, being a hemophiliac, or having muscular dystrophy. We don't inherit two X chromosomes, we only inherit one. There's no backup. So color blindness, muscular dystrophy here is tied into a gene called the dystrophin gene. Okay, the dominant allele is the normal allele that codes for the normal dystrophin protein, which helps stabilize your skeletal muscle cell structures. The recessive allele is the defective gene. Okay, it doesn't code for a functional dystrophin protein, so what you see in the offspring is muscle atrophy. The average lifespan for an offspring for a male, you primarily find it in males, okay, since it's an X-linked genetic disorder. And what happens is the average lifespan of a male used to be 5 years of age. I don't know about today, but it used to be 5 years of age. So, in other words, the male offspring wasn't going to be producing their own offspring. Now, and then there's hemophilia. This one by far was the most popular one. I first was introduced to this when I was in elementary school, and my friends and I used to say, "Why is it always the little boy that's our age that bleeds to death every time they show us a video on Helia?" Well, it's because it was an X-linked genetic disorder, and you primarily find it occurring in males. I'm sure it's happened out there, but I've never come across any information where a female has inherited hemophilia, but I'm sure it's out there, especially with improving treatment for symptoms. But this genetic disorder, which is an X-linked genetic disorder, okay, part of its popularity came from a royal family, Tsar Nicholas II of Russia. Okay, familiar with royalty. Okay, when the king and queen or the czar and his queen have a child, they'd like it to be a male to inherit the throne, and that was the case for Tsar Nicholas. Had a son, Alexis, but he had what they call in lay terms bleeder's disease, hemophilia. Had a cut, couldn't form a blood clot, bled to death. Well, later geneticists come along, managed to somehow, some way, I haven't looked it up, but managed to obtain DNA samples, and they create what we call a pedigree. They start with the oldest generation, Queen Victoria and her husband Albert, or not her husband Albert, or I should say husband Albert. But the pedigree necessarily doesn't have to do with the couples married. Okay, it just has to do with, hey, they had an offspring. So Queen Victoria and Albert have an offspring, Alice. Okay, what is not known at the time though is Queen Victoria is heterozygous dominant for hemophilia. On one of her two X chromosomes, she's carrying a mutated allele that does not code for a clotting factor, okay, required to form a blood clot. She happens to pass on the X chromosome with that mutated recessive allele onto her daughter. Albert did not have hemophilia. His X chromosome was carrying the dominant normal allele, and that's what he passed on. That's the only X chromosome he has to pass on to Alice. So Alice is a carrier like her mother. Alice and Louis, they have their own child. It's a daughter, Alexandra. Louis does not have hemophilia. On his one X chromosome, he has the normal dominant allele that he passes on to his daughter. Alice, however, happens to pass on the one of two X chromosomes that's carrying the recessive allele that causes hemophilia, and she passes it on to Alexandra. Tsar Nicholas, normal male, no hemophilia. The one X chromosome he has the normal dominant allele. Okay. He passes that on to, I'm sorry, but he has a son. Tsar Nicholas doesn't pass on his X chromosome with the normal allele. Since he has a son, that means Tsar Nicholas passed on his Y chromosome. Alexi's only X chromosome is coming from his mom, and it just so happens she passes on the X chromosome with the recessive allele that causes hemophilia, and thus eventually he ends up dying. Okay. What it was found is that on the X chromosome, it codes for one of several types of blood clotting factors. And whenever you get a cut, this triggers, what do you call it, a cascade of events or a chain of chemical reactions that requires several clotting factor proteins. And if one of those clotting factor proteins is not present, it cuts off that chain reaction and you can't form a blood clot. Okay, and this is what was happening here. So these are the three types of X-linked genetic disorders. What I want to go into next, I call chromosomal abnormalities. Okay, and I come at this from 2 points. The chromosomal abnormality could have something to do with the offspring not inheriting the normal number or the correct number of chromosomes. And again, we're going to focus on us. Okay, keep in mind, for the normal human being, our diploid cells should have 23 pairs of chromosomes for a total of 46. That's normal. Okay, well, what's been found is in some offspring, the offspring either inherit one too few chromosomes or one or more extra chromosomes. And we'll go over this process called aneuploidy. Then we're going to transition into a different set of causes of chromosomal abnormality where the DNA that forms the chromosome, something happens to where it's rearranged. Okay, and I'm going to show you a couple of disorders or diseases that are a result of these changes. So starting with A, chromosome abnormalities having to do with an alteration of chromosome numbers inherited by the offspring. This is called aneuploidy. Okay, aneuploidy, the way I define it, this is when an abnormal number of chromosomes within a gamete is passed on to an offspring. Look at these two karyotypes here. Okay, if you look at these two karyotypes closely, notice down here and notice over here. I've already shown you several normal karyotypes. If you look at this karyotype, notice the first 22 pairs of chromosomes, autosomal chromosomes, are normal. We have 22 pairs. But when you get to the 23rd, what should be a pair, we only have one X chromosome. This is a disorder, a type of aneuploidy that we call Turner syndrome. Only happens in females. I'll go over this more in a second. If you look at this karyotype, jump down to the sex chromosomes. Notice the pair of X chromosomes, so we know it's a female offspring. It's a pair. Everything's fine. Then we start scanning the first 22 pairs of autosomal chromosomes on the karyotype, and when we get to the 21st pair, we notice there's not a pair of chromosome 21s as there should be. There's a third one present. This is what most karyotypes look like for offsprings that have Down syndrome or trisomy 21. These are both types of aneuploidy. What causes aneuploidy? Well, aneuploidy is caused by non-disjunction. Okay. Again, you have to know chapter 13, cell division involving meiosis. If meiosis goes correctly, okay, then a process called disjunction will occur. Okay. Disjunction will happen in both meiosis 1 and meiosis 2. When disjunction happens in meiosis 1, our tetrads will be segregated into separate haploid cells. During meiosis 2, if disjunction occurs correctly, let me use this other one over here. If disjunction occurs correctly, okay, then what will happen is our sister chromatids, I'm going to go back, what will happen is our sister chromatids will be separated into separate haploid gamete cells. This is disjunction. Segregation and separation happen correctly. However, non-disjunction is the cause of aneuploidy. It can occur during meiosis 1 or meiosis 2. Okay, so they're showing you here on this side, on the left side, we have non-disjunction occurring during meiosis 1. Okay, one of the two tetrads is not segregated. Therefore, we do not transition from a diploid to a haploid state. Instead, what happens is we go from one diploid nucleus to two nuclei that are aneuploids. Okay, you can see here one is short, one of the sister chromatids, the other one has an extra. If non-disjunction occurs during meiosis 1, every gamete that is produced is not a haploid gamete. They're either carrying one too many chromosomes or they're carrying one too few. Okay? Therefore, if any of these gametes, which are not haploid, okay, are involved in fertilization, then we're going to end up with an offspring that if they survive past birth, the offspring is going to be an aneuploid, and it's just a matter of how they label, okay, the disorder that they have. Is it Turner syndrome? Is it Klinefelter syndrome? Is it Down syndrome? Over here, though, if you look, they're showing you non-disjunction if it occurs during meiosis 2. If non-disjunction occurs during meiosis 2, then what happens during meiosis 1 is we get segregation of our homologous pairs of sister chromatids, which we call tetrads, into separate cells to where when meiosis 1 ends, we've gone from diploid to haploid state. However, if non-disjunction occurs during meiosis 2, that means at least one of our sister chromatids is not separated into daughter chromosomes. So they're showing you here, when meiosis 2 occurs, we actually have two haploid cells carrying sister chromatids that need to be separated into daughter chromosomes. And it's showing here where one of the two, a problem is non-disjunction. This results in 50% of the gametes not being haploid, but instead 50% of the gametes are aneuploids. If you look in the other cell, they're showing you separation of sister chromatids into daughter chromosomes works out fine. And notice the other 50% of the gametes being produced, they're normal haploid gametes. But the problem is there's still a 50-50 chance, okay, that a gamete that is an aneuploid could be involved in fertilization, resulting in an offspring, okay, that has an abnormal number of chromosomes. It's non-disjunction that causes aneuploidy, okay, and then depending, okay, on where the incorrect number of chromosomes is occurring, all right, that will feed into how they diagnose that offspring that has an aneuploidy, such as the case with Klinefelter and Turner syndrome. Eventually, what was discovered is there were certain males, they tended to grow abnormally tall, but they were sterile. Okay, now there could be different causes for this, but one of the causes for this had to do with the male. Instead of inheriting one X chromosome, they would inherit one or more extra X chromosomes. Okay. Here's a karyotype of one of these male offsprings. They grew abnormally tall, but they were sterile, couldn't have their own children, and it just so happened that in their case, okay, the cause of this was due to a type of aneuploidy called Klinefelter syndrome. Klinefelter syndrome, two things, everybody. First of all, for Klinefelter syndrome, the offspring is always male. Please remember that, okay? Okay. So there's a type of aneuploidy that they call Klinefelter syndrome. The offspring is always male. And what's the cause of this syndrome? The male offspring has inherited one or more extra X chromosomes. In this case, it just so happens that the male offspring inherited as 3 extra X chromosomes due to non-disjunction. Okay, sometimes they'll only inherit 1 extra X chromosome, 2, but sometimes they'll inherit 3 extra X chromosomes. So the way we denote this in this case, okay, is what they'll do for Klinefelter syndrome. All right. Normal offspring should inherit 46 chromosomes. But if you look here, 47, 48, 49. So what they'll do is they'll denote it this way. They'll write 49, and I hope I can do this. Just messed up. And then what they'll do is, depending on how many extra X chromosomes the offspring is inherited, if you notice there are 4 X chromosomes, they'll write it down this way to denote, hey, this offspring, they're an aneuploid, they have Klinefelter syndrome, okay, meaning that they inherited extra X chromosomes, and if they inherited as many as 3 extra X chromosomes, they'll write down they don't have the normal 46 total chromosomes, they have 49, and then they'll let you know where the extra ones came from. It has to do with the sex. Turner syndrome only occurs in females. And what happens with Turner syndrome, in this case, is that the female offspring inherits only one X chromosome. So the way a geneticist will write it out is 45XO or X0. Okay, this over here indicates, hey, look, they didn't inherit a pair of sex chromosomes. They inherited one. Something went wrong. It was non-disjunction. So whenever you see a karyotype such as this and you come down to the 23rd pair and you notice it's just an X chromosome, there's only one, not two. You have to know two things. One, okay, the offspring is female. Okay. Two, the offspring only inherited one X chromosome instead of a pair. For those female offspring that are aneuploidy and diagnosed as having Turner syndrome, they tend to be sterile. They can't have their own children. Okay. Now, what they've actually discovered, okay, is that aneuploidy can happen, okay, with the various pairs of chromosomes that you see on a karyotype. It's just much of the time the offspring either doesn't survive to birth, there's a miscarriage, or they die shortly after birth. Okay, but these two here, okay, you hear about Klinefelter and Turner syndrome because there are offspring that have not only survived, birth, but they've lived. I mean, they've lived an extensive lifespan. One other thing with Turner syndrome is that secondary female characteristics, okay, I said they're sterile. Well, that's tied into secondary female characteristics. They don't fully develop during puberty. Now, sometimes aneuploidy doesn't occur with the sex chromosomes. Sometimes aneuploidy occurs with your autosomes. Okay, the most popular one, and excuse me for using popular, but it's the most common one, is Down syndrome, better known as trisomy 21 among geneticists. Okay, listen carefully. For this type of aneuploidy, it's occurring, okay, in one of the first 22 pairs of chromosomes, specifically 21. Therefore, the offspring could be male or could be female. Okay, the chromosomal abnormality or the aneuploidy is not happening with the sex chromosomes. It's happening with one of the 22 pairs of autosomal chromosomes. What should be the 21st pair, an extra one is inherited. So again, geneticists will call it trisomy 21. It's better known in the population as Down syndrome. Could be male, could be female. Now, what they found is with the increasing age of the mother, especially if the mother's capable of having children as she reaches an age of 40, maybe after, that increases the likelihood that non-disjunction will occur, and it'll occur here, okay, at 21. Now, why? I haven't seen any research, but I haven't looked for it either to see if they've uncovered that. All I can tell you, everybody, is our body's no different than the vehicles we drive. Okay, no matter how well you take care of it, the wear and tear eventually adds up. Okay, so certain processes that occurred normally with no problem, all of a sudden become a problem. Chromosomal abnormalities sometimes can occur when there's a rearrangement or a change in chromosome structure. Okay, so for this second cause of chromosomal abnormalities, we don't call it aneuploidy. Okay, it has to do with something occurs to the DNA that makes up a chromosome. Okay. 4 different processes can occur that results in some type of rearrangement in the DNA that forms a chromosome. Either some of the DNA that forms a chromosome is deleted, it's lost. So we call it a deletion. They're showing you, hey, we have DNA that forms a chromosome. The DNA makes up regions A through H, but for some reason, whether it's due to chemical exposure, mutagen or carcinogen, UV light, or maybe something happened during S phase, DNA replication during S phase, a region of DNA that makes up the chromosome is deleted. It's lost. And you can see they're showing you how that changes the structure. Okay. When a deletion occurs, normally it cannot be picked up on a karyotype unless it's a significant loss of DNA. Most of the time you can't see this. All right. Duplications. Duplications have been found to occur very commonly in our DNA. As a matter of fact, it's duplications, okay, that have been identified, okay, in particularly on the first 22 of 23 pairs of our chromosomes, okay. It's these duplications that occur in our chromosomes that geneticists eventually realized we can focus on these duplications, which happen to be unique to the different pairs of autosomal chromosomes, and use them to create a DNA fingerprint. So whenever you hear somebody talking about DNA fingerprinting, what they're actually identifying are different regions along your chromosomes where DNA has been duplicated. They're showing you here when a duplication occurs, again, for some reason, okay, what'll happen is we'll go from a chromosome that has regions of DNA that form A through H to where for some reason in this example, regions of DNA that forms regions B and C is duplicated. So now instead of B and C occurring just once, we have it repeating. What they'll do is they'll sequence the DNA and they'll say, hey, this DNA makes up region B, this DNA makes up region C, but then they'll notice that the sequence over here repeats over here. Sequence over here repeats over here. It's these duplications, okay, again, that geneticists realized we can target these duplications on different pairs of autosomal chromosomes and create a fingerprint to identify a person. Another process that can occur that can cause a rearrangement in the DNA that forms a chromosome is called an inversion. You don't hear much about these, but bottom line for an inversion is they're showing you here DNA that makes up regions A through H of the chromosome. And for some reason, okay, in this example, the DNA that makes up regions B, C, and D, okay, is cut out, flipped 180 degrees, and reinserted back into the same stretch. So the overall DNA that makes up the chromosome, or the overall length of the chromosome, doesn't change, but the order of the DNA has changed. It's been flipped 180 degrees. Whereas with the duplication, it depends on how many duplications you have. It may or may not be picked up on a karyotype. Normally, it's just picked up through DNA sequencing. Translocations. Translocations have to do with where a region of DNA on one chromosome is exchanged with regions of DNA on another chromosome that is not a homologous chromosome. So this is not crossing over. Translocation has to do with an exchange of DNA or DNA recombination that occurs between non-homologous chromosomes. This type of DNA rearrangement may or may not be picked up on a karyotype. So what they're showing you is for some reason this can occur. Again, it may be exposure to mutagens, carcinogens, UV light in the sun, or something haywire may have occurred during DNA replication of S phase. But what happens is that you get an exchange of DNA on one chromosome. It'll exchange its DNA with DNA on a non-homologous chromosome. They just exchange, they swap off, resulting in this here, and you can see how they label the regions. Okay. Now, sometimes when one of these processes occurs, it can lead, okay, either to a genetic disorder or some other type of ailment that is not considered a genetic disorder, but yet it has a negative impact on the phenotype of the offspring, okay, such as up here, deletions. There's a genetic disorder, okay, that is caused by an allele known as the TR gene. This gene is located on chromosome 7. Okay, so in other words, it's located on one of the autosomal chromosomes. But it is found to become a genetic disorder, and it was primarily traced back to those of European descent. And what they found is the most common cause of this genetic disorder, called cystic fibrosis, it resulted from a deletion of part of this gene. It's a very small deletion, so you can't pick it up on the karyotype, okay? The scientists would actually have to sequence the DNA that makes up the chromosome or sequence this gene to identify, okay, that it's a mutated version of this gene caused by a deletion. What they found is just a few DNA nucleotides, 3 of them are deleted from this gene, but it just so happens those 3 DNA nucleotides that make up that region of the gene that's deleted, they happen to code for a part of a protein that's responsible for binding to a type of energy that we talked about before, known as ATP. So what happens is in a person with cystic fibrosis, okay, the most common cause of this genetic disorder is a deletion that occurs in this gene. It's a very tiny deletion. But what ends up happening is mucus cells of the person with this genetic disorder, this genetic information is expressed, resulting in the production of a protein known as a chlorine ion channel. The problem is a small piece of this protein called the chlorine ion channel is missing, which they highlight here in red. That small piece that's missing in this protein is the part that happens to be responsible for binding to a type of energy known as ATP. So I kind of compare it to having a real nice car or truck or SUV, but the battery's missing. What good does it do you? So this is a case that happens here with cystic fibrosis. It's due to a small deletion. Another example of how a process changing or rearranging DNA that makes up a chromosome can lead to adverse effects on the offspring's phenotype has to do with the translocation. Okay, it was discovered there's a type of cancer called chronic myelogenous leukemia, more popularly known as CML. This type of cancer results in cancerous white blood cells that do not fully develop. It's a malignant type of cancer. Okay, so it's terminal, as they call it. It does lead to death. So when scientists discovered this type of cancer and called it CML or chronic myelogenous leukemia, they wanted to look for the cause. And they found out that the cause of this type of cancer, okay, resulted from a translocation between chromosomes 9 and 22. What they found is that roughly in here and right in this region here, what they found is that when a translocation happens between chromosome 9 and chromosome 22, which they're highlighting the parts of DNA that are exchanged, this region here on chromosome 9 ends up next to this region here on chromosome 22, which is right down here. Just putting a little bracket. They're trying to put a little bracket. So this region right here ends up next to this region on chromosome 22 here, which I tried to put a little bracket. And what they've discovered is that due to this translocation, due to this exchange of DNA between chromosome 9 and chromosome 22, that region right here of DNA that ends up directly next to this region of DNA forming chromosome 22 results in the formation of a cancer-causing gene that causes CML. Okay, and they discovered that it was due to a translocation between 9 and 22. The amount of DNA that is exchanged or translocated between these two chromosomes can be picked up sometimes on a karyotype because it's a significant amount of DNA that is exchanged between the two. So it can be picked up. Now, what they overall have found out is that these processes can occur and we never know it. The reason for that is only 1.5% of our DNA contains genetic information. Much of our DNA is what at one time was described as junk DNA, but now they backed off of calling it junk DNA because they find it has a lot of other uses. But it's possible that a mutation or caused by one of these processes occurs and we never know it. Okay. Now I didn't mention it, but for duplications are not only the focus for DNA fingerprinting, but they are tied into some other types of disorders or diseases such as Fragile X syndrome and Huntington's disease. Okay, but I just didn't mention it here, and I'm not going to test you on this. Okay, so I'm going to stop the recording.